VIASURE Real-Time PCR Detection Kits. VIASURE Real-Time PCR Detection Kits allow the specific identification of pathogens in biological samples in all stages of infection. It is based on the real-time PCR technique, an in vitro technique used to amplify specific DNA fragments and detect its accumulation in real time by measuring the increase in a fluorescence signal. To begin the reaction, the temperature is raised to 95 degrees. At this temperature, the double-stranded DNA is separated into single strands. Then, three major steps are repeated during each cycle. First, denaturation to obtain single DNA strands. Second, annealing. The temperature is then lowered to approximately 60 degrees. This allows the primers and the fluorescent probe to bind to your target sequence of interest. And third, extension. Between 60 and 72 degrees, the polymerase extends the primers and cleaves the probe, which generates an increase in the fluorescence signal proportional to the amount of amplified product. After one cycle of PCR, there are twice copies of the target DNA. These steps are repeated through around 45 cycles. When the starting material is RNA, one first additional cycle of reverse transcription is required. The real-time PCR reaction requires the following components: Taq DNA polymerase, reverse transcriptase, primers, probe, DN-TPS, MgCl2, magnesium chloride, buffer, stabilizers, internal control. VIASURE Real-Time PCR Detection Kit contains the following components: well strips with all the previous reactives in a stabilized format, positive and negative control, rehydration buffer to reconstitute the stabilized product, water RNA SE DNA SE free, optical caps for sealing wells during thermal cycling. Molecular diagnosis unit consists of 4 areas: nucleic acids extraction, pre-PCR, setup, PCR, amplification and results interpretation. The first area aims to process potentially infectious samples and subsequent extraction and purification of nucleic acids with the RNA-DNA VIASURE extraction kit. After extraction, the sample is ready for the next area. Take the VIASURE Pathogen Real-Time PCR Detection Kit and separate the required number of wells, including samples and 2 reserved wells, for the positive and negative controls that must always be included in each assay. If necessary, cut the required number of wells with a scissors and remove the protective aluminium seal. Reconstitute each one of the wells with 15 microliters of the blue vial rehydration buffer. Finally, add 5 microliters of the purple vial negative control in the reserved well for negative control. After this process, move the rehydrated well to the next area. 2 independent processes are made in the third area. First, the positive control rehydration. VIASURE Pathogen Real-Time PCR Detection Kit contains a positive control that must be rehydrated before using this assay. It is recommended to open and manipulate before starting with the assay. Reconstitute the red vial positive control with 100 microliters of the white vial RNase DNase free water and mix with the help of the vortex. Store at -20 degrees after resuspension and divide in aliquots to minimize the freeze-thaw cycles. The second process is the addition of the nucleic acids and positive control to the rehydrated wells. Add 5 microliters of extracted DNA, RNA from each sample in the rehydrated wells, with the exception of the 2 well reserved for the positive and negative control. Add 5 microliters of reconstituted red vial positive control in the reserved well for positive control. To finish, divide the required optical caps number and close the wells. this process, move the plate to the real-time PCR equipment. Place the plate in the thermocycler, program the thermocycler and start protocol. When finished, analyze the results with the software installed in the equipment. VIASURE provides maximum flexibility and compatibility with leading open system thermal cyclers and the latest technology for the stabilization of products. Ready-to-use kits with all the necessary components for the testing. Long-term stability that facilitates transport at room temperature. Easy to use. It minimizes the number of manipulations, reducing time and possible errors. Versatile. Possibility to analyze from 1 up to 96 samples per testing. Exogenous internal control. Evaluated and validated according to ISO 13485 and CE marked. VIASURE Real-Time PCR Detection Kits.